Good afternoon, everyone, and welcome to today's EMSL Learn webinar on PFLOTRAN, Subsurface Reactive Transport Simulator. And we are so glad to see you today. The EMSL Learn webinar series is designed to help provide information to the scientific community on resources available at EMSL for research in the biological, environmental, and computing sciences. Our webinars are posted in the EMSL Learn section of the EMSL website. And so we invite you to visit that section to see our past webinars. Our next EMSL Learn webinar is on April 23rd, and that one will focus on EMSL's user portal, now called Science Central. It's a one-stop shop for users. More information will be provided through our social media channels, via email, about all of our upcoming events and opportunities. So make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. And go ahead and take a look in the chat. You'll see the links provided there for you. Okay, so today's presenters include Glenn Hammond, a computational geohydrologist and senior research scientist at Pacific Northwest National Laboratory, who will introduce PFLOTRAN and discuss how to use the code to simulate subsurface reactive transport processes. And our next speaker will be Katie Muller, also a PNNL Earth scientist, who will present on how to incorporate soil organic carbon processes into PFLOTRAN. Now, there will be time at the end of today's webinar for questions. So we encourage you to post your questions in the Zoom Q&A chat area, and so that our speakers can answer them during the Q&A. We won't be answering them during their presentations, but it will be at the end. We have about 15 minutes for Q&A. Okay, so with that, we will begin with Glenn.
Welcome to this webinar. It's an opportunity for us to introduce PFLOTRAN. I'm going to describe some applications of PFLOTRAN. We probably won't go into great detail as far as how you run PFLOTRAN or set up input files and so forth. But first, we want to poll the audience. We want to see what your background is. And so can we put up that first poll? This is going to help me tailor this a little bit to the audience too. So I'm going to, I'm going to begin with a discussion of how we use modeling to simulate subsurface processes, provide a little bit of background into PFLOTRAN, its history, and then I have actually 6 example problems that I'm going to show. Okay, so it looks like we have quite a few modelers and experimentalists here, and some developers. That's where I would put myself. So, all right, thank you for responding to that poll. So, what I have here is a representation of the subsurface. We have our soil, we have grass at the top. Let me see if I can get a pointer here. And just a little bit of delay. Sorry. Try that again. Can I get the pointer for laser pointer? Well, we'll just proceed. All right, so we have our grass here at the top of the domain. This is where we're going to have organic matter. So you see that there's leaf matter in there. You're probably going to have roots that are in the top layer of soil. As there's rainfall depicted by the arrows, water will flow down through the domain. Okay, and it's gonna flow through a tortuous path around the soil grains. As that water flows past the organic matter at the top of the domain, there is organic matter that dissolved into the water. There's also particular organic matter that will travel with the water, and that travels down through the soil also. So what we do is when we model, we discretize this domain. So we break it up into grid cells, and we model the flow and the transport through those grid cells. So let's look at that in more detail. So here I have a single grid cell down the middle of the domain. we have fluxes. The blue arrows depict water fluxes. The orange arrows depict solute fluxes in between the grid cells. Within the grid cell, we're going to have chemical calculations. So we have chemistry, and the circular pattern here just means we're constantly calculating chemistry within that grid cell. Throughout the entire domain, we're calculating the chemistry at all grid cells, not just these 4, but at every grid cell in this domain. I think there's 36 cells here. And so when we're running reactive transport, we're modeling the infiltration of the water, the transport of the solutes, the organic carbon coming down from the top, and the chemical reactions that are occurring in each grid cell. Okay, so let's move on to a quick introduction to PFLOTRAN. PFLOTRAN is a petascale reactive multi-phase flow and transport code. By petascale, I mean it's been run on at least 100,000 processor cores. So it runs on a supercomputer. It also runs on a laptop computer, so it runs on that span of machines. It's open source. It has a GNU LGPL license, meaning you can add proprietary code to PFLOTRAN, but it has to be a separate library or a wrapper code. Any modifications to the original source code itself must be freely available to the public. It's written in modern Fortran, object-oriented Fortran, and from that, what I mean by that is we use pointers to procedures in Fortran classes extensively throughout the code. It's founded on a large number of well-known and supported codes or libraries, PETSI being our most important library. And we've demonstrated performance on very large problems composed of up to 3 billion degrees of freedom or unknowns, and on around 262,000 processor cores. Most of the time, you're going to run PFLOTRAN in parallel on hundreds to thousands of processor cores. It scales well to about 10,000. Beyond that, the scalability tails off. And you can see that here in this plot here. The blue line, we're starting at 1,024 cores or processes, and as we go, the blue line represents linear performance. So as you double the number of processes, the time is cut in half, and the red line is the performance of PFLOTRAN out to 27,000 processes. So we're very pleased with this. Okay. So a little bit of history on PFLOTRAN. It originates from my mentor, Peter Lichtner. He had a code called FLOTRAN. And as part of my doctoral research, working with Peter, we developed PFLOTRAN. And so you can see below the timeline there, these are the various developers that have been involved in PFLOTRAN development. And above that are the various institutions that have had a say in its development, had people work on it. These are some pictures of a number of the developers. I would say 2/3 of these developers are actively participating in the development of PFLOTRAN today, including a few of these that are international. Joe here is in the UK, same with Paulo. And then Moise is from France, but he lives in Montreal, Canada. And so this is our developer crew. So physical and chemical process models that are simulated by PFLOTRAN. So we simulate variably saturated groundwater flow, multi-phase flow, both miscible and immiscible. We handle heat transfer, multi-component transport, multi-phase transport. So we can do gas transport and chemical reactions in that phase. And then biogeochemical reaction, along with geomechanics and geophysics. So the highlighted capabilities here, I'm actually going to show examples later on in this presentation. I have one slide on numerical methods for all the numerical methods enthusiasts out there. For spatial discretization, we use the finite volume approach using the 2-point flux approximation. We support structured and unstructured grids. On the right-hand side, you can see an unstructured grid that's actually used for discrete fracture networks. This was developed by C.T. Shkara years ago. Our time discretization is fully implicit backward Euler. For the nonlinear solver, we use Newton-Raphson. For the linear solvers, our default is by CG-STAB and then LU for direct solves. Our coupling of physics, and I'm going to focus on the physics that we're going to discuss today, that is flow and transport. Flow and transport are predominantly sequentially coupled, but there are a few flow modes where we have them tightly coupled. For transport and reaction, we can use sequential coupling, that's often referred to as operator splitting, or the global implicit approach, that's fully coupled. We tend to run with global implicit most often because it's very robust and it performs quite well, minimizes the error. And when you can hop on a supercomputer, you can solve those very large systems of equations. Our approach to parallelization is through domain decomposition, and I can answer more questions about all these things if people want to ask at the end. Okay, let's discuss reactive transport processes that are supported. So for transport, it's multi-component, meaning you have multiple chemical species. They can be in the mobile or immobile phases. So think of soil organic matter. That may be an immobile species. We support advection and hydrodynamic dispersion. So advection is where a solute is transported with the flow of water, and the hydrodynamic dispersion is both mechanical mixing and molecular diffusion. On the reaction side of the code, we support aqueous speciation. You can think of that as equilibrium reactions, say the carbonate species, carbonate, bicarbonate, and aqueous CO2. Well, there's equilibration that occurs between all those species. Aqueous speciation calculates this. We support microbiological reactions. So we focus on Michaelis-Menten kinetics, or think of that as being multiplicative Minot or dual Minot. We support biomass, and then we have inhibition terms. So under certain conditions in the subsurface, microbial reactions will not proceed. Denitrification will not occur in oxidizing conditions, and so, or aerobic conditions. And so we inhibit the ability of a denitrification reaction to occur in say, the vadostone. And I'll show an example. We support mineral precipitation dissolution. We follow the transition state theory approach. And there are a number of capabilities that I will demonstrate in a copper leaching example. We support radioactive decay and ingrowth. For absorption, there is a large suite of absorption models that we support. Historically, the code, we performed quite a bit of research 15 years ago using surface complexation at the Hanford 300 area. That's just north of our site here in Richland, Washington. And we model equilibrium, kinetic, and multi-rate surface complexation. But we also have isotherms for the engineers out there that want to try the isotherms that they learn in grad school. And then we have the reaction sandbox and the soil carbon sandbox. Katie is going to discuss the soil carbon sandbox and some of the quick soil carbon processes that are implemented, that can be implemented in it. The idea with the soil carbon sandbox is you can define soil carbon pools and the mass transfer processes between the pools. And we kind of tailored that sandbox to be more user-friendly to domain scientists or let's say experimentalists. So, soil carbon, PFLOTRAN's internal units are moles per liter water, right? And soil carbon tends to be in grams per kilogram soil. And so we do the conversion internally so that the user can think of things in their own space. Going back to the reaction sandbox, I'm going to discuss this a little bit more, but this is a modular framework for defining kinetically formulated reactions within PFLOTRAN. Okay, I want to pop up the second poll question. We just want to find out if people have used any or would be using any of that in any of their research. So we should have a poll question number 2 coming up here. In the meantime, I'm going to see, I guess I can't. Okay, while you're answering that poll question, I'm just going to discuss our main governing equation. This is the advection dispersion reaction equation. What we have here is the left-hand side, we have the accumulation of mass over time. We have our advection and dispersion, that's our Darcy flow velocity vector. And this is our dispersion term, our hydrodynamic dispersion term that factors in tortuosity, liquid saturation, and porosity. We have a source term right here, and then this summation here is over kinetic reactions. Our equilibrium speciation is embedded in this capital psi term right here. This is basically where we sum the free ion concentration that I see right there, plus its stoichiometric contribution to all the secondary aqueous complexes. Now that's kind of a complex statement, but this is our equilibrium constant. We have a activity coefficient, and then we have our species concentrations. And so using this algebraic equation, we embed this algebraic equation in the governing equation and we solve for it. And that's how we solve for our equilibrium chemistry. For more information on this, I could spend 2 hours. I can honestly spend a week talking about this. So I would refer to Peter Lichtner's publication down here for more details. So let's discuss the reaction sandbox for a second. This is a methanogenesis reaction where we have hydrogen combining with CO2 producing methane, okay? There are a number of ways to define the rate expression for this. We could use a second order rate expression, which is the function of a rate constant times hydrogen and CO2 concentrations. We could use multiplicative Minot, where we introduce half saturation coefficients for each species. We could add biomass, that's this X right here. There may be a case where we need a more specialized function. The first three are supported in PFLOTRAN. The bottom rate expression is not. This is where we introduce a Hill function, where we raise the concentration of CO2 and its half saturation coefficient to an exponent n. To implement this, you would have to go into the code and modify it. And so the challenge here is, how do you go into PFLOTRAN if you're new to the code? How can you modify it? This originates from a conversation I had with Hari Rajaram, a professor I had years ago, where he came up to me and he said, how can I help my graduate students get into the code and implement their own rate expressions? And so that's where the reaction sandbox came to be. So if we were to open up the source code and look at it, it would look like this to most people. It would be confusing. There would be a lot of jargon, and it just would not make a whole lot of sense initially. It would take time to understand what the code does. So what we've done is we've cordoned off a section of the code where we pass select state variables. In this case, it's hydrogen and CO2. We pass these into our sandbox. Inside the sandbox, you can write a small subset of code to calculate this rate expression. The rate internally in PFLOTRAN will be in moles per second. That's passed back to our residual function. That's part of our Newton-Raphson method. And then we are off and running. So if we allow the user to define their own rate expression, they are able to add their contributions to the code. There's more detail on this in this geoscientific model development paper. I'd invite you to download it. It's out there, it's free, if you want to learn more about that. The reaction sandbox has been used extensively at PNNL, but I would also highlight that a researcher, Ben Tutelo, out of the University of Calgary, has used this extensively in his research. And Ben simulates mineral dissolution, precipitation reactions, and he often steps outside transition state theory for defining those rate expressions. So there's a number of papers that he's produced on it using the reaction sandbox. So he's a very adept user. Okay, let's discuss the 6 scenarios that I'm going to illustrate. These are just high-level presentation here. I'm just going to show some movies of some simulation results. The first one's rainwater infiltrating a heterogeneous porous medium. Then we have a tracer injection in a different porous medium. We have gas injection. We have mineralization of organic carbon and oxic groundwater, in situ copper leaching, and then a denitrification problem. So let's move on to the first one. In this scenario, we have layered heterogeneous media. So we actually have 2 layers. You probably can't see it very well, but there's a line right there. We have an upper porous medium and a lower porous medium. They're heterogeneous, but they have the mean value in the lower porous medium is lower. So you're going to see water flow in between these 2 lines. It will pool slightly on the top of the interface between the layers, and then it will flow down to the saturated zone. So let's run this. Pools right there, and then you see how it flows down to the saturated zone. So this is just an illustration of flow within PFLOTRAN, variably saturated flow in PFLOTRAN, and how it looks over time. Let me run it one more time. Just go back. You can see it flowing in, pooling, and the effects of the heterogeneity. There's kind of preferential flow through this region right here, where there's higher permeability. Okay. So our next example is tracer injection in a doublet. This is actually a short course problem from the PFLOTRAN short course, where we inject water through a well and we extract water down gradient. We have an upland boundary condition, and water regional flow is toward the river to the right. You have a vadose zone, and then the blue is saturated groundwater. So I'm gonna inject tracer into the groundwater, and then we're going to watch it flow through the porous medium. And so what we see here, what you see right now is the heterogeneous permeability field. And as I start the movie, you'll see the tracer flow in right here and then flow down gradient. So there's a tracer flowing in. These isosurfaces are at 0.1, 0.5, and 0.9 tracer concentration. This is normalized tracer concentration. So let me start that over again. So you can see it flowing through a tortuous path due to the heterogeneous permeability, flowing out down the plane. So there we have a solute transport combined with flow. Our next example is where we have a gas injection scenario. This is where we're going to inject gas. There will be multi-phase flow. So I'm going to inject gas in the lower left-hand corner. There are these low permeability clay lenses that are in a sand. And you'll see how it progresses up to the outlet at the upper right over time. There is the lower lens. It hits the other two lenses and then flows out of the domain. And you'll see that over time, it actually pulls up quite a bit at the top boundary. And that's a no-flow boundary along the top here. It's completely no-flow. Okay, so this is an example of multi-phase flow. There are no chemical reactions in this other than the dissolution of the gas, or the injection of the gas and then the bubbling of the gas. Here we have an example of a flow field. We have a regional flow field going from upper right to lower left. And we have an oxygenated domain. That's, you see right here, we have a large oxygen concentration. We're going to inject organic carbon into these 4 wells right here. And you're going to see that organic carbon react with the oxygen. We have organic carbon plus oxygen produces carbon dioxide and water. There will be a buildup of carbon dioxide in the domain and the subsequent precipitation of calcite, a mineral. Now you're going to see the plumes kind of wave like a flag. That's because this regional flow, I've assigned it so that it varies over time just a little bit. And so let's watch that. There's the injection, the consumption of oxygen. Oxygen is the acceptor here, and the production of the aqueous CO2. And then we have the precipitation of the calcite down here. So this is an example of a microbially mediated reaction. We're using multiplicative Minot. There is no inhibition here, but we do use multiplicative Minot to simulate this. Let me run that one more time. Just you watch. So we have the injection of the donor. There's the acceptor of the oxygen, the inorganic carbon that's produced, and the calcite production. So if you're thinking of sequestering carbon in the subsurface, this is how you could mineralize CO2 in the subsurface. And there are ideas of doing this in agricultural areas. Okay, this next example is of in situ copper leaching. So we can inject lixiviant into the subsurface. Lixiviant is something that's used to dissolve a mineral and extract critical minerals or metals. In this case, we're extracting copper. So we have a heterogeneous permeability and porosity field. We have a regional gradient here too. Our magenta wells, these are production wells. The white wells are injection wells. So we inject, and these 4 wells contribute to this production well. These 4 wells contribute to this production well, and so forth. So there are 9 production and I think 16 injection wells. What more can I tell you about this? It's 8 meters thick, so this is a 2-dimensional solution. And so our chemistry here, our primary species, we have 12 of them. Coppers are our metal of interest. And then we're using sulfuric acid. It's actually 1 mole sulfuric acid that's being injected in the subsurface. And so we're going to monitor the sulfate concentration that results from that. Now, you do not want the sulfuric acid to travel off-site. We want to keep it contained. We want to extract it through the production wells. And so that's something that we're going to look at here. Is we don't want excursions where it leaves the site. We have a large number of secondary aqueous complexes in this problem. That's the equilibrium chemistry I was discussing. And then we have a number of minerals. Chrysocolla is the copper mineral that we will be dissolving. And this is the rate expression. This is transition state theory, where we have our specific surface area, our rate constant. This is the prefactor, where we take the hydrogen ion concentration, raise it to a power. And then this is our affinity factor. So when this is equal to 0, we're in equilibrium, the rate is 0. And then when it's supersaturated or undersaturated, it causes the rate to run in different directions, so forth, so that you have precipitation or dissolution. So here when I start this, we're gonna see on the left-hand side, you're gonna see the chrysocolla. It's going to dissolve, so it's gonna turn blue inside of the dissolution area. And then you're gonna see the sulfate injection on the right-hand side. So sulfate's being injected, and then the mineral starts dissolving. Continues to dissolve. And then it's completely dissolved here. Okay, let me run that one more time. Sulfate's being injected, or the sulfuric acid is being injected, and the chrysocolla is being dissolved. You'll notice these, in between these production wells, there's a section where the chrysocolla is not being dissolved as quickly. And that there is our optimization schemes you could use on the pumping scenario to improve the extraction over time. Okay, and a point I want to make here is that if you see the white between these production wells, this is chrysocolla that has not been extracted, whereas closer to the injection well, there's much more dissolution. So you could change your pumping strategies to maybe inject here and extract over here, and that will pull water across that section right there and remove the mineral. You'll see also that we have excursions here. So you see that we have sulfate or sulfuric acid essentially exiting the domain. This is problematic for mining operations. This is very concerning. And so what I show on the next slide is that you can actually adjust the pumping in these outer injection wells. We can take some of the mass from the lower left injection well and these other two down here. We can redistribute it to other wells, and you can actually improve or decrease the amount of sulfuric acid that exits the domain. And so this refers to lixiviant, but that's the sulfuric acid. The blue line is the mass of sulfuric acid that's exiting the domain with the original scenario. And you can see that as we increase the reinjection, the amount that's reinjected and put in other wells, or we redistribute it, you can see that we decrease the mass that leaves the domain. You'll also notice that we actually improve the amount of metal that's extracted. So the amount of copper that's extracted here actually goes up ever so slightly because of our improved pumping scenarios. So here's where we can use reactive transport in a mining operation to inform decisions that are being made and improve the productivity and decrease the environmental impact. That's very important. Okay, my last scenario. This is denitrification under anoxic reducing conditions. So we have this. This is a vertical cross section. We're going to have water flowing in. We have a regional gradient, so water is going to flow to the right. In the vadose zone, it's oxidizing, and then in the saturated zone, you'll see that there are reducing conditions there. I'm going to release some organic carbon and some nitrate. You can think of various situations in the world where there are operations that may release nitrate into the subsurface, and it will show denitrification occurring. These are our microbially mediated reactions, and in this case, there is inhibition. So the middle nitrate reduction right here cannot occur until the oxygen concentration is below a certain threshold concentration. Same thing goes for nitrite. Nitrite cannot be reduced until the nitrate is below a certain threshold concentration. And so, all right, there's a lot going on here. We have this is just our groundwater saturation here. You'll see our oxygen concentration here, our organic concentration. We have a source tracer that's going to come in through the source zone. And then we have our nitrate, nitrite, and the N2, and then our initial tracer, and this is going to wash out in the vadose zone. Let's watch that over time. You can see our organic carbon coming in with that tracer. The nitrate comes in. You'll see that nitrate reduction does not occur until just about we're at the water table right there. So all through here, oxygen is being consumed to degrade the organic carbon. The organic carbon is the donor. It's the donor for all three of these, oxygen, nitrate, and nitrite. And you'll see that nitrite degrades down to the nitrogen. Run that one more time. Organic carbon reacts with the nitrate to produce a nitrite right there, and the nitrite reacts with the organic carbon to produce the aqueous nitrogen right there, and it flows down gradient offsite. So everything I've shown thus far has been in the aqueous phase, except for maybe some biomass in these problems. Next up, Katie is going to discuss how we can incorporate soil organic carbon and other organic carbon species into PFLOTRAN. Katie, I'll turn it over to you.
Thanks, Glenn. Let me take control here. Okay, I think I've got control. Let me just verify. Hold on, sorry everyone. Okay, got control now. So this section's more tailored towards the experimentalists, and I'm really going to be talking about reactions, about how biogeochemical reactions fit in, how we can use experimental data to build and implement reaction networks, and then eventually incorporate those within a larger PFLOTRAN model. So as Glenn illustrated, PFLOTRAN has a wide range of native capabilities. So that includes different flow conditions. So we can simulate 0D batch experiments all the way up to a 3D system, both with saturated and variable saturated flow. There's also extensive aqueous geochemistry that's harnessed in the code through pairing to thermodynamic databases. And then, of course, we have our biogeochemical reactions as well. So while the standard reactions in PFLOTRAN are very powerful on their own, there's always specific cases that require some level of customization, particularly on the reaction side. So to do this, we can use the reaction sandbox that Glenn discussed to define and implement our custom reaction networks. And those fit right into the native PFLOTRAN capabilities of flow and transport. So for example, we've developed a reaction sandbox recently tailored towards soil processes. And then we've also created a reaction sandbox for organic carbon speciation as well. So I'm going to walk through a few of these examples, but really the most important piece I want to really emphasize here is that the reaction sandbox can be used to capture just about any reaction or process of interest. So that's really, the important part. So let me just walk through how this process works more generally. Sorry. Having troubles advancing. There we go. Okay, so in order to build any reactive transport model, you first need to start with a conceptual model. So in a conceptual model is really a general idea of what's happening in your system. It identifies those governing processes. And I really like to think of your conceptual model as like a whiteboard drawing. Where are your main processes? You have those all sketched out, and then you have arrows showing how the system components are interconnected. So once you have your conceptual model, you need to translate that your system into mathematical expressions. And for the reactions, this results in the development of your reaction network. So your reaction network might only require native capabilities, or perhaps you need a reaction sandbox to implement more custom expressions. So from here, data is used to parameterize your model. And this is typically done with a parameter estimation tool that can be easily wrapped around PFLOTRAN. And this ultimately leads to a very useful parameterized, potentially predictive model. All right, so that was a little abstract. So now I'm going to talk through a pretty basic example kind of showing this process. So let's say you're a scientist and you're wanting to understand more about the soil carbon processes at your specific site. So the first thing you need to do is develop your conceptual model. So this is done by defining the different carbon pools of interest. So for this example, we've split the carbon pools into 5 different categories. So we have particulate organic carbon, mineral associated organic matter, dissolved organic carbon, and then CO2. So from here, we can then add those arrows to describe how the mass transfers between these pools and how they interact with each other to create this overall simple system. From here, we translate this conceptual model into mathematical expressions to create our reaction network as follows. And so here, we're keeping it really simple for this example. So all these reactions are assumed to be first order rate expressions proceeding at different rates from K1 to K4 as you go from the first to the fourth reaction here. So now that we have a simple model, if we put in initial concentrations for our various species of interest, as well as assigning representative kinetic rate parameters, even without data, we can run some simple informative simulations where we track the temporal evolution of all of our species. So for instance, this simulation can provide insight into how the mass transfers from POC over to mineral associated organic matter, DOC, and ultimately gets respired as CO2 over time. And so once you have a simple model, you can start playing some theoretical simulation games. So for instance, let's find out what will happen in the system if you increase your POC concentration initially. So we can do that just by changing that initial concentration, which you can see in the table, but also visually on the plot as well. And you can see that the increase in POC ultimately affects those intermediate species as well as the overall CO2 production curve. And we can do the same things with the rates. Let's say you believe that one of your reaction rates is actually a lot faster. You can test that out with a simulation. And in this case, you can see that the POC being converted happens much more rapidly and what that would do ultimately to your system. So even without detailed data to create a parameterized model, you can have these simple simulations that are valuable in terms of providing insight and enhanced understanding of your system. They can be helpful for identifying your controlling processes or perhaps even to inform your experimental design or sampling strategies to most effectively capture those system dynamics. All right, so that was a pretty simplistic model that we just walked through, but I do want to highlight that with PFLOTRAN, it is very easy to layer on additional complexity of interest. So in that last example, we only ran through respiration, aerobic respiration. However, this reaction network can be easily expanded. So for instance, we can, oh, I gave away the answer there. We can go through all the way through methanogenesis if we want to include those processes in our system. We can also include mineral dissolution precipitation reactions by adding those on, perhaps to describe that mineral associated organic matter in a more sophisticated way, and so on and so forth. And so really the point here is that you can layer on these levels of complexity within these reactions, all within PFLOTRAN. All right, and so all of this has been focused on the reactions themselves. However, really the powerful thing here about these simulators is that everything we've shown on the reaction side and creating those custom reaction networks can be applied to all sorts of different flow scenarios or domains, like what Glenn was stepping through before. So you can really take what you've learned about the reactions, maybe from a batch experiment, and you can apply those to more realistic scenarios. So for instance, maybe a 3D flowing system, and again, all within PFLOTRAN framework. Okay, so now I'm going to run through 2 real examples. So in this first example, we're using data collected from soil incubations. And one of the main questions that these researchers were interested in was understanding the impact of root loadings on CO2 production. So what they did was they ran a series of incubations where water and soil and roots were added and then left to incubate for 120 days. And really the neat part about these experiments is that the roots were isotopically labeled, which essentially allowed for us to independently track the contributions from roots and soil independently on CO2 emissions. And from the this experimental setup, we developed this custom reaction network shown here that consists of 4 reactions. The first 2 show the transformation from insoluble carbon, both from the soil and the roots, over to soluble DOC. And then the third and fourth reaction show that DOC is being respired to CO2. So using the data that was collected during their experiments, we were able to fit rate parameters to those 4 expressions, as well as some microbial biomass parameters as well, since these are all microbially mediated processes. We did this using PEST, which is a well-established parameter estimation tool that again just wraps around PFLOTRAN. And because these experiments were conducted using multiple root loadings, the parameters were first fit to this lowest root addition. So essentially we calibrated to this one experiment. And then we could turn around and use those fitted rates in a more predictive way to predict what the CO2 gas generation would be when you had a much higher root loading added to these incubations. So if the researchers in this case were interested, they could take this parameterized reaction network and actually kind of scale it up to a more realistic environmental setting, maybe running 2D or 3D simulations, perhaps with variable saturated flow conditions as well. And again, this can all be done within PFLOTRAN itself. So the next example is also for incubation. So these are incubations done for coastal wetland soils, and they were completed under both aerobic and anaerobic conditions, where CO2 and methane was production was measured over time. They collected aqueous samples temporally and analyzed them for a whole bunch of different parameters, but most important for the modeling efforts was they measured total carbon amounts, but also they looked at the carbon speciation using FTIC-RMS. So for these, we first built our most basic conceptual model of the system, where carbon is assumed to release from the soil into the aqueous phase, where it's assumed to be this generic carbon of CH2O. And then that soluble carbon is respired to CO2 over time. And from this conceptual model, we wrote our reaction network consisting of these 2 expressions here. And using the measured values from the experiments and the known applied conditions, we were able to model and fit some parameters to the experimental CO2 production. So from here, the next step was to figure out how we could actually utilize that speciated organic carbon data gathered from FTICRMS so that we could add that into our conceptual understanding of the system. So in order to do this, we built a reaction sandbox and a corresponding workflow within PFLOTRAN. And so this workflow takes our FTICRMS signal here that identifies thousands of individual carbon species in a single sample. And it creates a distribution based on thermodynamics of all of those different compounds. We then turn around and take that distribution and create a unique reaction network based on that FTICR-MS signal. So here it's showing this reaction network that consists of 10 different reactions based on a specific carbon species in each of those bins and the resulting stoichiometric coefficients to produce this overall reaction network. So from there, we can use that unique reaction network and perform reactive transport simulations with PFLOTRAN. And so with this approach, each of the carbon bins is tracked independently as a specific species. And then we can add them together and we can look at total carbon or aqueous chemistry, and of course the CO2 production as well. So from here, you can see that we've updated our original conceptual model. So now, instead of using that bulk carbon, we are informing that as a speciated carbon from the FTICRMS data. So our reaction network gets updated. So instead of a single reaction for aerobic respiration, we now have that 10 reaction network in its place. And we can run with this model in the same way we did before. You can see that we do we're able to provide a better prediction of our CO2 generation data using this carbon speciation data collected from FTICRMS. Okay, so we went through some general information about PFLOTRAN and some examples today, but there's a lot more information about PFLOTRAN that we weren't able to cover. So if you want to learn more, you can visit pflotran.org. We're happy to take your questions now, but if you do have additional questions, you can always email the Google group listed here with general questions, or you can email Glenn or myself with more specific questions. But before we open it up to Q&A, we really want to hear from you. So we would love to hear how you might use reactive transport simulations in your research. So if you don't mind, just take a moment and answer this poll question or put your response in the chat, and then we'll head over to the Q&A. Thanks.
All right, we're getting some great answers coming through. As folks are finishing that up, why don't we start the Q&A right now? Looks like a good number of people have answered thus far. So to kick off the Q&A section, Kadir is asking, carbon speciation was implemented in K-base before. Does it have standalone implementation or PFLOTRAN integration now? So I open that up to Glenn or Katie.
Katie can answer that one.
Yeah, I can answer that. Yeah, it does have standalone implementation. We have a paper that was recently published, and it's all up on I think a GitHub site with all the code. So you can go look there, but yes, it is standalone, and it's built kind of built upon the K-base idea, but it's inherently coupled into PFLOTRAN now.
Cool.
Is it,
Okay.
that's with the Lambda sandbox, correct?
Correct.
Yeah.
Okay. Mark is asking, transport of PFAS in the vadose zone has been shown to be strongly influenced by adsorption at air-water interfaces. Can you model that currently?
It depends what the process you're trying to model. I mean, we can model absorption, but there may be a specific equation, the approach that you want to model the absorption. I need to see the equation before I can answer that. I mean, if you just want to model it through a linear Freundlich isotherm, most certainly we can do it. Yeah. I'm always hesitant to be, well, you have to look at the equations of what people want.
Yeah. For large-scale CO2 sequestration modeling, which one do you recommend, PFLOTRAN or STOMP, and why?
PFLOTRAN. Because we developed PFLOTRAN. Recommend PFLOTRAN. If you want to work with us, I recommend using PFLOTRAN. If you want to work with the STOMP developers, you're welcome to go that route too. I will point out that we have implemented in PFLOTRAN STOMP's implementation of carbon sequestration, not carbon, the supercritical CO2 capability that's in STOMP has been migrated to PFLOTRAN over the past year, so it's available.
All right. Next one, for someone starting with an existing groundwater flow model simulation, what are the primary steps to incorporate reactive transport using PFLOTRAN?
It's a great question. I always advocate starting off simple. So if you're looking to model flow and reactive transport, start off with your flow simulation, get that up and running. Add tracer transport to it. So you're going to incorporate the transport process model with a single tracer and the boundary conditions for that. And then from there, I would encourage, it depends on the level of complexity in the reactive transport. If you're going to model mineral precipitation dissolution, I would encourage you to use the calcite. There's a short course problem on calcite dissolution. I would pull that chemistry in, and then from there, go more complex in the chemistry. But you want to do it in stages. If you do it all at once, you're going to get buried in complexity, and it's going to be very difficult.
Mm-hmm. Okay, so that was from Miguel, and Miguel has a follow-up question too. What are good starting points for working with the reaction sandbox? For example, demos or example problems?
So there's the paper that's out there that you can download from Geoscientific Model Development. With that, there are some examples that you can pick up. In the PFLOTRAN repository are some example implementations of the reaction sandbox that you use as a template. You begin from there and then you build on that. That's where most all of our users start. So it's actually in the regression test. I could point you to the exact one, but I can't right now because I can't post it to this screen. So, but if you reach out, we can share that with you. Yeah, it's a great question.
Yeah, I would just add, I think reaching out to us to get started on, what you need customization of is probably also a good route.
Good point.
And I have to do a mea culpa here. I said that was Miguel's follow-up question, and it wasn't. It was somebody else's question. Miguel's actual follow-up question was, for someone new to PFLOTRAN, but familiar with groundwater modeling, what is the typical learning curve?
If you've used other simulators, it should be fairly intuitive. I mean, I'm always open for feedback, but it should be fairly intuitive. What I would encourage you to do is go run. There's a variably saturated flow problem in the short course. Go look at the input deck, see what makes sense and what doesn't make sense, and start asking questions. Yeah.
Okay. Next question, Hansel asks, let's see, for what is the state of PFLOTRAN's GPU support?
We currently do not support GPUs. The back end of PFLOTRAN, the PETSI library, can leverage the GPUs, but none of PFLOTRAN has been refactored to employ GPUs. That may change in the future, but right now we have not done nothing.
Okay. Next question, mine asks, can you provide the basic tips briefly for building a RTM for predicting metal transport and speciation?
The basic steps, we would need to know more regarding your conceptual model, but that copper leaching scenario that I presented is very similar to what you're going to want to accomplish. And so you could lift the chemistry from that example. You don't have to have the complex flow. You could create a 1D column and put that copper leaching scenario in there. Actually, that's the same problem I used for my master's thesis, and just model it. And it will, you can learn from that. So again, I would encourage you to reach out, share your conceptual model if you're willing to share it, because there's a lot of detail in that. But it's always good to pick up an existing example and build from that.
Mm-hmm.
And I will add, in terms of the chemistry side, it is always helpful to identify what your most important species are outside of PFLOTRAN ahead of time, using, Geochemist Workbench or Freak-C in order to know what species you want to model in PFLOTRAN, because we don't pick those for you, you define them.
Mm-hmm. Okay. We have 2 minutes left, so we've got a few more questions here. One just popped in. Where do researchers usually run PFLOTRAN simulations? On a supercomputer?
I would say most people run it on their laptop. And then when you want to go to the large scale, you want to run large, when you go over hundreds of thousands of grid cells in 3D, that's when you're going to migrate to a supercomputer. Yeah.
Mm-hmm. Okay, let's do one more question, and then I believe you have one last poll question. Is that correct? Okay, because we're down to 2 minutes. So last audience question, is the lambda binning available in the sandbox?
Yeah. Yep. We built a sandbox just for the lambda binning.
Okay.
So Katie, does it perform the binning for you, or do you have to do that externally?
It does it in the Python script before. So I guess it's not within the sandbox, but it feeds directly into into the sandbox.
And that's covered in the
point.
paper, right?
It is covered in the paper, yeah.
Yeah. All right. So before everybody leaves, Carly, can we get that last poll question up? And folks, if you can answer this one last poll question before you head out, we would greatly appreciate it. We thank you so much for joining us today to learn more about PFLOTRAN, and we're all looking forward to working with you in enhancing your research in subsurface flow and transport. So thank you, everyone. And don't forget, April 23rd is our next webinar on Science Central.